Yeah, welcome to the morning session. It's my pleasure to start today with the virtual reality to immersive analytics in bioinformatics section. So what's the basic idea behind this? So I think in bioinformatics, and especially in structural biology, 3D stereoscopic displays are used since a very long time. So already around the years 2000, people were using the stereoscopic displays, like for example, CRT displays. And so we came up with the idea, let's make a new special session, which is taking a look backward, what was done so far, and in addition, what are the new approaches which we want to address today. So, and we organized the session together with Mark Baden, so maybe just say hello, yeah. So then Michael Krone, who is sitting there, and of course Andrew Woods, who also supported us a lot. So, and we will start today with the first talk. So it's my pleasure to invite Hua Wong. So he's from the Department of Biology, Biochemistry and Environment at University of Rennes, Champagne-Ardenne. And his research topics are structural biology, molecular biology, and computer graphics. So it's a very interesting combination of research topics here in the context of this conference. And his most recent publication is on molecular simulations of elastin and elastin-like peptides, past, present, and future. So we are looking now forwards to his talk, which is entitled Mesoscopic rigid body modeling of the extracellular matrix self-assembly. Please start.
Thanks for the introduction. So I'd like to first thank the organizer for inviting me to present my talk, and the audience for being here so early in the morning. Today I'm going to talk about the work we did on the extracellular matrix. So the first part of this talk, I will explain a little bit how we build our model. And in the second part of the talk, I will present a little bit what we do with interactions and visualization of the model. So first, why are we interested in the basement membrane? The basement membrane is part of the extracellular matrix. It is the part of the extracellular matrix that physically link cells to the extracellular matrix. So it has an important function. The problem occurs when there are some dysfunction in the basement membrane. It can lead to some serious diseases. So I illustrated this with this example. In the case of cancer, you have in situ cancer that is still quite easy to treat by surgery. And when these cancerous cells manage to find a way to bypass the basement membrane, we evolve into what we call invasive cancer, which is generally not a good news. So perhaps even for this reason alone, it would be interesting to study the structure and the behavior of the basement membrane. However, the basement membrane is a very difficult biological object to study, because most of the components of the basement membrane are located at the mesoscopic scale. So what does it mean? Objects that are at the mesoscopic scale are too small to be observed directly with a microscope, but they are still too big to be resolved with classical structural resolving methods like X-ray crystallography or NMR. However, that doesn't mean we don't have any observation at this scale. We indeed have data from atomic force microscopy, or we have envelopes from cryo-EM or SACS. We even have rotary shadowing imaging from electron microscopy. But generally, these data provide only blurry or partial information. That means we can have good information on the aspect or the size of the molecule, but any atomic detail is completely lost. And even if we had, by magic means, a mesoscope to observe directly this structure, we are talking about something that is extremely dense. This is a very thin part of the extracellular matrix, and in this extremely thin region we have molecules such as the nitrogen, the perlecan, the laminin, type 4 collagen, and so on, all stacked into a very small space. However, we managed to use the game engine Unity to develop tools that allows us to study such mesoscopic systems. So what you see in the video, it is a typical simulation environment that we have at the moment. So as you see, we put some molecules. So these are simplified representation of the components that make up the basement membrane. So once we have our simulation set up, we can let it run. So in the following slides, I'm going to explain to you how we managed to get to this result. So the first stage of our work is to rebuild the molecular models. For this, we have to integrate many data from many various sources. So I'm going to present you the example of the molecule perlecan. So the very first information we get when we search for information, we search the literature. So what does the literature tell us about the perlecan? The perlecan is a linear protein that is composed of 5 multi-domain, labeled 5, 4, 3, 2, 1, and each of these multi-domain is composed of smaller domains. So the thing is, this is like having the blueprint of a molecule. Because the thing is, we don't have any all-atom structure of the perlecan because it is simply too big to be resolved. However, if we look each of the individual domains of the protein, we find that they have been resolved and can be found online on the protein data banks. So for example, we have the structure of the heparan sulfate, of the SEA module, and so on. So once we manage to recover all these little domains, what we can do is simply assemble them by following the blueprints. For this task, we are using the 3D modeling software Blender. The 3D modeling software Blender allows us to manipulate the 3D structural data. So we can simply position the small domains for example, to create the chains of heparan sulfate. We can also modify or change the position or the orientation of the various domains that make up the perlecan. So it's not an obligation to use Blender, but it's a free software, so why not? So this is what we get from rebuilding the perlecan molecule. And what we can see is there is a qualitative agreement between the structure we've reconstructed and atomic force microscopy data for the perlecan. So for example, we have an agreement on the size. The size aspect is respected. We can see these 3 little domains that also appear in this picture. So that means we are probably doing something right. So if we apply this approach to all the other molecules of the basement membrane, so this is what we obtain. We apply the same method to get the collagen 4, the nidogen, the integrin, laminin. And what struck us the most, the first time we saw this, all these molecules together, is the sheer scale of these molecules. What you see on the right is how the biologists generally picture this molecule. And for example, the perlecan is often pictured as a small molecule. And if you look at it from real scale, it's a rather huge molecule. So once we have all the models for all the components of the basement membrane, we have to make them move. For this, we are going to use rigid body dynamics. So the first question we have to ask ourselves is, why don't we use all-atom molecular dynamics? After all, they are large systems like the HIV capsids that have been modeled and simulated in all-atom simulations. So the HIV capsid is made up of 64 million atoms. By comparison, the pelican I've shown you has 63,000 atoms only. But the thing is, if you want to make an all-atom simulation of the HIV capsid, for example, they made simulation of 100 nanoseconds, they have to use the computing power of this machine, so the Blue Water supercomputer that runs at 13 petaflops. So as a comparison, it's roughly equivalent to 7,000 MacBook Pros. So this illustrates the amount of computational power that is required for all-atom dynamics. And if you look on the right, it is a picture that I like to use in many of the presentations I do on the basement membrane, because it is called basement membrane by David Goodsell. And what you can see is that if we want to model this kind of system in all-atom, in its all-atom glory, we are not only talking about one pelican, but about several pelicans. And this applies to all the other molecules that are involved in the basement membrane. So, if we wanted to do a similar simulation with a reasonable timescale that gives us some information, for example, for hundreds of nanoseconds, if we wanted to do this in all atoms dynamics, we wouldn't, even a 13 petaflops computer might not be enough. Any petascale computing, it would not be enough. So, to model this kind of system, we have to find a way to simplify this. So one of the sources of inspiration we have is CellPack. So CellPack has been developed in the Johnson lab at UCSF and the molecular graphic labs at the Scripps Institute. And what CellPack does is it creates mesoscale molecular models from given molecular ingredients. And it does so by searching the optimal packing from these molecular ingredients to fit inside this container. The things that interest us in CellPack is the fact that the molecular ingredients are treated as rigid bodies. That means they don't take into account all the atoms in the molecular ingredients. They consider them as a whole, a shape that doesn't change shape. And by doing so, they are saving a lot of computing resources. From their paper about CellPack, they say that a laptop has enough computing power to create such a mesoscale molecular model. The other source of inspiration we have for our work is my own work I did on the yeast cell nucleus. So in this work, we have modeled the chromosomes of the baker's yeast using chains of rigid bodies. We use this model to successfully study the self-organization of the yeast. And the idea there is to simply fuse the two concepts together. So instead of having just rigid molecular ingredients and rigid bodies chains on one side, we would like to have rigid molecular ingredients chains. And as you are going to see from the next slides, it's not completely it's not as far-fetched as it seems. Because in fact, biologists have already think of complex molecules as groups of domains. So I've already introduced the case of the pelican. On the left, you have the integrin, that is a molecule that is on the surface of the membrane. And you see that it is composed of, it is defined as a group of domain. So you have the beta propler, the tie attached to each other, and the same thing for this chain. So the question you probably have at the moment is, what is a rigid body? So the idea I would like to, it doesn't have to be something abstract or complex. Rigid bodies, in our case, are molecular domains. The main difference is that these molecular domains, in order to be used in a physics engine, we add some physical properties to these domains, such as the position or the orientation, and we also add a collider for collision management. And also, we are not only using rigid bodies, we are using, more precisely, articulated rigid bodies. What does it mean? It means that we have rigid bodies, just like in CellPack, but each of the rigid bodies are attached by a small joint. I'm not going to enter into the details of what a joint is. All you have to know is that it is a very strong positional constraint that links the bodies together. So this is what, okay, 5 minutes, I'm almost done. So this is what it gives on the Pelican in motion. So right now we are using Langevin dynamics. Basically, Langevin dynamics is Brownian motion with some terms to imitate the viscosity of a fluid. And as you see, the physics engine only has a couple of rigid bodies to deal with. So we save a lot of computing power that we can repurpose for more fun applications. So, for example, we attached virtual reality headset and controllers on our simulation. The goal here is to make our simulation more immersive and interactive. For this, we use the virtual reality toolkit, mainly for two options. The first one on the picture on the left is the one that allows the user to be immersed and move inside the simulation. So for example, in here, we are just teleporting to a pad that gets us closer to the simulation. And the other type of functionality we use from VRTK is the ability to interact with the object inside our simulation. So here you see a controller that meets an object, and we can manipulate it directly. So I would like to conclude this talk by showing you this video. So if this was an all-atom model, there would be roughly 500,000 atoms. It would be difficult to do molecular dynamics on such a large system. But with a rigid body representation, the physics engine only has to manage roughly a couple of hundred rigid bodies. And as a matter of fact, this sort of simulation can run on the laptop I am currently doing my presentation with. It's a Surface Pro 4, and it's not even the top of the line. It's the Core M3. It's really the baseline Surface Pro. So this also gives me the opportunity to tell you, if you want to see a demonstration, we will show the simulation this afternoon. I'm right, huh? It's this afternoon. So if you want to see it in action, you can come and see us. That being said, I thank you for your attention.